Hello everyone, my name is Nelly Nyaga and I would like to present the results of my study entitled Investigation of the Cardioprotective Effects of the Freeze-dried Extracts of Salvia coccinea Juss ex mure in the isolated heart in Sprague-Dawley rats using a global ischemia model. The supervisors for this study were Dr. Peter Waweru and Dr. Frederick Bukachi. So what is coronary heart disease? Coronary heart disease is among the leading causes of death and disability worldwide, with data from WHO stating that up to 12% of deaths worldwide are due to coronary heart disease. The most severe manifestation of coronary heart disease is myocardial infarction, which occurs due to ischemia, and reperfusion injury, which occurs during coronary heart disease. So what is this ischemia? Ischemia is an imbalance between the oxygen demand and the oxygen supply in the tissues. So now the remedy to this imbalance is prompt reperfusion, which is actually a double-edged sword because it causes reperfusion injury. So when you talk about ischemia being destructive, this is exactly what happens in ischemia. So first you have the decreased oxygen supply compared to the demand. This leads to an increase in the hydrogen ions inside the cell, the acidity or acidemia inside the cell. This activates a channel we call the sodium hydrogen exchanger, which actually now leads to an increase in the sodium current inside the cell. This increase in sodium concentration inside the cell leads to the activation of the calcium sodium exchanger, which pumps out the sodium in exchange for the calcium, leading to a calcium overload inside the cell. And this calcium overload is the destructive point because calcium is involved in activation of many enzymes, is involved in the it leads to now overactivation of the cardiomyocytes, which leads to hypercontracture, which leads to cell death. So during reperfusion now, after ischemia, what happens is we restore the oxygen that was insufficient. So we get a burst of reactive oxygen species, which lead to the opening of a pore on the mitochondria called the mitochondrial permeability transition pore. So this pore worsens the calcium oscillations that were present during ischemia. Okay, so then we have more calcium released from the sarcoplasmic reticulum, leading to hypercontracture, and then we get sarcolemma rupture and cell death. Also, we have the pH normalizes but leads to calpain activation, calpain activation is also caused by the calcium. And calpains are enzymes that are involved in activation of apoptosis, which is programmed cell death, and this will eventually lead to cell death. So this brings us to cardioprotection. Here is where now we protect the cell, the myocardial cells, from this ischemia reperfusion injury that will occur during coronary heart disease. So basically, cardioprotection involves all the mechanisms that are aimed at preventing cell damage that occurs during acute cell injury. It may be done either by myocardial conditioning or pharmacological intervention. So in myocardial conditioning, we basically prime the heart. The heart is exposed to brief periods of ischemia, that are not damaging enough, but it just primes the heart, makes it strong enough in case it encounters a bigger attack. In pharmacological conditioning, what happens is after an ischemic attack, you now give a pharmacological agent which prevents further damage. And this is actually what is applied clinically because a patient will present with a myocardial infarction, then you give an agent that will prevent further damage. So the pathways that are involved in cardioprotection are several, and many agents have been implicated in this process, but there are two major ones that have been implicated, and these are the adenosine pathway and the opioid pathway. And these two are linked to the nitric oxide pathway, which leads to the activation of protein kinase G, and the endpoint is the inhibition of the opening of the mitochondrial permeability transition pore, which we saw is the endpoint in causing cell death that occurs during ischemia reperfusion. So these two pathways act to inhibit opening of that pore and therefore leading to cardioprotection. So now we come to Salvia coccinea, the hub in this study. It's commonly referred to as scarlet sage, and it belongs to the Laminaceae family. It has been used in China after boiling it in water to bathe varicosities, blood clots, and congested blood. Phytochemical studies on it have revealed the presence of salvianolic acids, which have actually been shown to be active antioxidants. The rationale of this study is that one, coronary heart disease is the largest cause of mortality and morbidity worldwide. Two, there is no effective therapy or treatment to ischemia reperfusion injury to date. And thirdly, Salvia murugi, a herb in the same family with Salvia coccinea, has been shown to exert cardioprotective effects on ischemia reperfusion injury. The research question was, do the freeze-dried extracts of Salvia coccinea have significant cardioprotective effects against ischemia reperfusion injury? The hypothesis, the null, was that the freeze-dried extracts of Salvia coccinea do not have significant cardioprotective effects against ischemia reperfusion injury. The alternative hypothesis was that the freeze-dried extracts of Salvia coccinea have a significant cardioprotective effect against ischemia reperfusion injury. The general objective of the study was to investigate the cardioprotective effects of Salvia coccinea against ischemia reperfusion injury and to determine its mode of. The specific objectives were: 1. To investigate the effects of the freeze-dried extracts of Salvia coccinea on the left ventricular developed pressure, on the maximum rate of contraction, or the maximum rate of relaxation, and on the heart rate. 2. To evaluate the effects of the freeze-dried extracts of Salvia coccinea on the myocardial structure through a histological study. And 3. To elucidate the possible mechanisms of the cardioprotective effects of Salvia coccinea. The experimental design. The study was carried out in 2 phases. The first phase involved the cardioprotection experiment. There were 5 groups, each with 6 rats. The first group was the control group, which was where the hearts were perfused with plain Krebs-Hensley buffer. The second group was the positive control group, where the hearts were perfused using glucosamine and non-cardioprotective agent. There were 3 test groups, where one was perfused with 50 milligrams dose of Salvia coccinea, group 2 with 100 milligrams Salvia coccinea, and the third 200 milligrams of Salvia coccinea. The second set was the mode of action experiment. There were 3 groups, each with 6 rats. Group 1 was reperfused using theophylline together with 50 milligrams Salvia coccinea. Group 2 was reperfused using naloxone together with Salvia coccinea, and the control group hearts were reperfused using Krebs-Hensley buffer and Salvia coccinea. The materials and methods. The experimental animals were 42 adult male Sprague-Dawley rats between 250 and 400 grams, and the equipment was a Lagendorf apparatus, which is shown in the diagram. Next. The procedure. The animals were weighed, anesthetized, and heparinized according to their weights. The heart was then excised and an aortic cannula inserted into the aorta. Perfusion with Krebs-Hensley buffer was then initiated at 8 to 10 milliliters per minute. A balloon was then inserted in the left ventricle and inflated, and then it was connected to a pressure transducer, and through it, the monitoring of the pressure was started. After that, the hearts were let to equilibrate for 20 minutes, after which they were exposed to 30 minutes of global ischemia by halting of the perfusion. After the 30 minutes of global ischemia, the hearts were then exposed to 90 minutes of reperfusion with the respective treatment depending on the group their heart was in. The histological examination was performed on the isolated heart after the functional study using the Mason's trichrome staining technique, and it was viewed under the light microscope. The features that were assessed include structure disruption, foci of necrosis, macrophage infiltration, and interstitial edema. The histological grading system that was used was based on a study conducted by Philho et al. And there were 4 grades used: 0, which indicated no change; 1, which was mild focal myocardial cell damage; 2, which represented moderate extensive myofibril disintegration; and 3, which was severe, representing necrosis with diffuse inflammation. The data, all values were expressed as mean plus or minus the standard error of mean. The data were analyzed using one-way ANOVA and post-hoc Tukey HST. Kruskal-Wallis and Dunn's post-hoc test were used for the histological damage using Graf-Bud Prism 6, and the results were considered statistically significant only if the p-value was less than 0.05. Ethical approval was sought from the Biosafety, Animal Use and Ethics Committee, Faculty of Veterinary Medicine, University of Nairobi, and all the animals were handled according to the guidelines provided, which were in accordance with the PHLASA guidelines of Guillain in 2012. The variables measured included, the direct measurement was the left ventricular developed pressure, and from this, heart rate, the maximum rate of contraction, the maximum rate of relaxation, and the histological evaluation were also assessed. So the results. So this first set of graphs represent the left ventricular developed pressure in millimeters mercury. So graph A represents the equilibration values in all groups, all the 5 groups. And as you can see, in the equilibration, there was no significant difference of the left ventricular developed pressure across the groups. So group B represents the left ventricular developed pressure after ischemia, when now the reperfusion with the respective agent was initiated. It is evident to note that the group reperfused with 50 milligrams of Salvia coccinea, the percentage recovery was significant compared to that of the control group, which was perfused only Krebs Hensleit. It was even more than the 100 milligram group and even more than the 200 milligram group. Also, graph C, the late recovery, the left ventricular developed pressure was highly significant compared to the control group and the 200 milligrams group. The second set of graphs represents the maximum rate of contraction, which is an indication of the contractility of the heart. Again, graph A shows that the equilibration values were not different across all groups. Graph B shows that the 50 milligram group recovered significantly more than the glucosamine group, which is actually a non-cardioprotective agent, and the 100 milligram group. Also in the late recovery, the recovery was not significant, but you can see that the 50 milligram group recovered higher than the glucosamine and all the other groups. Results. Results 3. This set of graphs represents the maximum rate of relaxation, which is an indication of the diastolic ability of the heart. The ability of the heart to relax. So again, graph A, the equilibration values were not different across the groups. In graph B, the 50 milligram group, the percentage recovery of this group was significantly higher than all other groups. And the same applied in late recovery, where also the 50 milligram group recovered highly compared to all other groups. To the heart rate, the equilibration values were not significantly different across all the groups. In graph B, we see that if you look at the glucosamine group and the KH group, you'll note that during reperfusion, the heart rate increased. And in the heart treated with Salvia coccinea, you'll note that after reperfusion, the heart rates go down in both the early phase of reperfusion and the late phase of reperfusion. A clear indication that the freeze-dried extracts of Salvia coccinea lead to a negative chronotropic effect. They decrease the heart. Okay, so now this set of graphs represent the percentage recovery of the different variables after blocker treatment. In the left ventricular developed pressure, we see that the percentage recovery of the 50 milligram group was significantly higher than of the naloxone group. So it's evident that naloxone completely abolished all the protection afforded by the 50 milligram dose. On the maximum rate of contraction, we see that there was no significant difference across the groups, but it's evident to note that the 50 milligram group recovered to a higher extent compared to all the other groups. On the maximum rate of relaxation, we see that the 50 milligram group recovery was significantly higher than the naloxone group, and also the naloxone group recovery was significantly lower than the theophylline group. On the heart rate, we see that the percentage recovery in the naloxone group actually led to an increase in heart rate compared to the 50 milligram group, where we see that the heart rate actually reduced. Also, the theophylline group, we had a reduction in heart rate, and the control group, the Krebs-Henslate group, had an increase in heart rate. The histological study. So here these results show, these are photos indicating the foci of necrosis. So the Salvia 50 milligram group only has one foci of necrosis. Looking at the 100 milligram group, the foci of necrosis increases, and even compared to the Krebs-Henslate and the glucosamine group. But you see that the 50 milligram group has the least foci of necrosis. This set of photos shows the structure. The 50 milligram group, you see, has the most preserved cardiac structure compared to the 100 milligram group, the 200, and the glucosamine group. So the results of this study are actually consistent with previous published studies on the cardioprotective effects of other Salvia species, and it also corroborates with a study that was done to investigate the cardioprotective effects of Salvia milrogiza, where they actually suggested a lower dose than the 100 milligram dose. And in this study, we used the 50 milligram dose, which was actually the most. Salvia coccinea was actually advantageous compared to other cardioprotective agents, in the sense that in this study, the maximum rate of relaxation recovered to a higher level compared to other studies where it recovered to lower levels. Also, compared to the study done on Salvia milrogiza, the heart rate in that study remained constant, whereas in this study, the heart rate became lower. And it has been shown that negative chronotropic effects are actually protective in myocardial infarction. Also, Salvia coccinea showed potency in both the early phase of reperfusion and the late phase of reperfusion, in contrary to other cardioprotective agents which remain potent only in the early phase of reperfusion. Late phase of reperfusion. There are, however, some limitations to this study, including that the metabolic substrates that are usually utilized by the heart, including lactate and free fatty acids, were absent in the buffer, and the fact that we lack the normal humoral and extrinsic neuronal regulation of the heart because we use the isolated model. However, the fact that we used the heart in isolation helped us to deduce the exact findings without the influence of the entire body. In summary, the results of this study show that the aqueous extracts of Salvia coccinea have significant cardioprotective effects against ischemia reperfusion injury in both the early and late phases of reperfusion. In this study also, the functional results agree with the histology results, where we see that the 50 milligram dose is the most potent dose in both phases, higher than both control groups. This study also reveals that the extracts of Salvia coccinea have negative chronotropic effects, which are actually protective. And finally, that the cardioprotective effects of Salvia coccinea are mediated through the opioid and adenosine pathway, and majorly through the opioid pathway, because you see that when using naloxone, the effects of the 50 milligram dose are completely abolished. In conclusion, These findings indicate that Salvia coccinea is a potential therapeutic agent against ischemia reperfusion injury, and we recommend that further advanced research be carried out to determine the individual components of the aqueous extract that is cardioprotective. Thank you very much.